So, welcome to the opening lecture of the eHorizons conference. I think we're all in for a great treat this evening because we have three superb talks. And I'd like to thank you very much for coming. So first, a little bit of background. The James Martin School was created in June 2005 from a large endowment from James Martin. It's a unique collaborative effort, and its goal is to formulate new concepts, policies and technologies that will make the future a better place. And within this, there are 10 institutes, and one of the institutes is the eHorizons Institute, which is a collaboration between the Oxford eResearch Centre, which is actually housed in the building you're in tonight, and the Oxford Internet Institute. One of the main activities of the eHorizons Institute is to produce a new publication called the Worldwide Science, the Promises, Threats and Realities of E-Research. So tonight we're looking at e-research. What is it? The best definition that I've come across is used in Australia. It's e-research encapsulates research activities that use a spectrum of advanced ICT capabilities and embraces new research methodologies. It's often used interchangeably with e-infrastructure and cyber infrastructure, but with an emphasis on the research which happens as a result of the infrastructure. And it's one of the issues we'll be discussing tomorrow at our conference, really what we mean by e-research, and is it distinct from e-infrastructure and cyber infrastructure. Some people call it research in an exponential world. So we have three leading e-research case studies that we're going to look at tonight from Denis Noble, Martin Kemp, and Jonathan Zittrain. And it's a chance to learn about the new opportunities for research underway in this university. There are three case studies which will be available for inclusion in our book. And it's a chance to really understand, as the speakers give their talks, which aspects of the research are groundbreaking, moving into new areas. How is the research held together in this collaborative activity? What have been the limitations for further progress, and which directions are being pursued in the future? The three talks are, well, in fact, the remit we gave the three speakers was through their own work to explain how e-research enables new forms of collaboration, visualization, and data collection in sciences and humanities. And then we left them to decide the titles of the talks. So without further ado, I'm going to hand straight over to Denis to start with the first talk.
Thank you very much, Paul. Does somebody need to switch over? Yes, it looks as though it's already in position. Interdisciplinary is certainly the term you would use of the work I'm going to describe to you today. There will be, as you will see, clearly some biology involved. I am a biologist, a physiologist. There will be some mathematics, some computing, some engineering, some physics, some chemistry. And if you have a look at this book, which sums up quite a lot of the background to this kind of work, you'll find that there's also linguistics and philosophy. What I'm trying to do is to unravel complexity in biological systems, and that's why I need interdisciplinary work in order to achieve that. The problem is that we have now, as a consequence of around 30 or 40 years of what I would call reductionist research in biology, unraveled many of the processes and components involved in biological systems, all the way from genes through to the whole organism, and indeed higher still to the environment. And I've drawn arrows here that we might describe as the reductionist view of biology. That is, that genes code for proteins which form pathways which then interact to form subcellular processes which then interact to form cellular processes, and all the way up. The dilemma is that that's not the way it works. There's also feedback down from all kinds of levels, with systems levels triggering processes at lower levels, various processes controlling, and you'll notice I've switched this arrow around, the expression of genes. That's done by protein machinery, and all levels can also mark the genome by processes called epigenetic marking. And so the problem is to try to unravel all of that, and you really can't go from the bottom upwards. Just two calculations that will illustrate that. The possible number of combinations of interactions of 25,000 genes, which is the present number thought to be present in the human genome, is of the order of 10 to the 70,000. There wouldn't be enough material in the whole universe for evolution to have explored all the possible interactions that there could be, even over the billions of years of the evolutionary process on Earth. So if biology couldn't have done it, evolution couldn't have done it over those billions of years, we're certainly not going to be able to do it. Another calculation, a single cell. Represent all the molecular processes in a single cell by quantum mechanical computations, you'd require something like 10 to the 27 blue genes. That is the computer that IBM built at around $100 million. There wouldn't be enough material in the whole solar system to build that many computers. So what we do is to try and use insight at all kinds of levels, working through these feedbacks from the different levels down and up, to try and understand processes first at the cardiac cell level, and then, as I shall show, to go up higher. And the work began a long time ago. I was asked once how many people have contributed to this work. I would guess at least 200 people, and many of them are situated right down there in New Zealand. Some are over in the United States, and that means obviously that we've clearly had to use e-connection, internet, and other forms of video conferencing quite a lot. But the work began way back in 1960 when I first started to analyze the flow of electric current through processes that we now know are proteins in the cell membrane. And what we found in the experiments done just over 40 years ago was that if you study the flow of potassium current through a cell membrane, this is its electric potential, this is the current flowing through it, there's clearly two components there. I won't bore you with the technical detail of the biology, but it's fairly obvious there are two components. There's one component that switches off as you change the potential in that direction, and there's another one that switches on. And they do so at different speeds, and we know what those speeds are roughly. This one is very fast, this one is very slow. And that was the basis, those are the two arrows, the fast and slow arrows. And that was the basis of putting a computer model together way back in 1960. This was the first computer model of a biological cell after the modeling of the nerve cell by Hodgkin and Huxley, and it was based to some extent on their work. Particularly in representing a process called a sodium channel. I used their equations. I then set up equations for these two potassium currents that we discovered, and for good measure there's another process in there too, but I won't talk about that. How to compute it. That led to having a set of differential equations, of course. There was a computer in the laboratory. It was the Brunsviga, and you see, of course, a model of that in the computing laboratory, the one used by Leslie Fox, I think. Andrew Huxley, to solve this equation, took something like 6 months to reconstruct the activity of a single cell. In fact, any given computation that he did took him to reproduce 5 milliseconds of a nerve impulse activity. It took him about 8 hours. Well, heart cells are much slower than that. 500 milliseconds is the minimum you need. That would take about 3 months of doing this. I once played table tennis with Andrew Huxley. He beat me hollow, and I think his biceps had clearly benefited from this. There were, of course, valve computers in those days. The one I used was Mercury. I also used Atlas up in Manchester. Mercury was in London. This actually, I don't have a photograph of either of those. If anybody in the computing lab has photographs of those machines, I'd love to have them. This is a similar machine. It was EDSAC, used by Andrew Huxley in Cambridge. And, of course, we played around with the machine code of those days. I even made the computer sing songs. We had it singing Song of the Volga Boatman when it was all working well, and oh dear, what can the matter be when it wasn't working well? Anyway, 2 or 3 months later, there were a couple of nice papers in Nature reconstructing for the first time the rhythmic activity of a cardiac cell. Obviously, the main interest here is, can you reproduce the fact that there's rhythm in the heart? The rhythm doesn't depend on the nervous system telling it what to do incidentally, though the nervous system modulates it. And the answer is, yes, you could. I won't go through all the technical details of that. Let's try and go on to what we do today. What we do today is to measure from real cells with very, very fine probes. This is an experiment from one of my former students, David Eisner, who's now a chairholder in Manchester. If we can get that movie to work. Yes, there it goes. The cell will contract, as you can see, but at the same time as it is moving, we're recording with a microelectrode some of the electric current flowing through those protein channels, many different protein channels. And in fact, today, to reconstruct the activity of a cardiac cell, we need many, many more protein channels, many more other proteins that are called carriers, which we didn't know about way back in 1960. We model intracellular events. Again, I won't bore you with the full detail of that. And for the drug industry, we also model a large number of different receptors. This, of course, is a cartoon diagram, but all of those components are represented by differential equations of one kind or another, differential and algebraic equations. Let's make one of the more modern models run. This is a model of pacemaker activity in the sinus node. That's the pacemaker region of a rabbit, so it's beating rather faster than my heart. There's 4 beats per second. Your and my heart's beating away at about 1 per second. And there are 3 of those components, those molecular components shown here, also oscillating away at the same time. And when I showed this a number of years ago to a group of newspaper science journalists in London, one of them said to me, well, Dennis, that's very nice. You've reconstructed beautifully the rhythm, the electrical changes during the rhythm of the heart, and if I superimposed a real recording of an experiment on that trace, you wouldn't see the difference. You can surely now, therefore, tell us which is the gene, which is the protein that generates cardiac rhythm. What is the cardiac rhythm gene? I had to reply to him, well, there's no such thing. Well, there must be. You've shown at least some of them here oscillating away. Surely one of them is driving that process. So no, it's not. Remember those arrows that I showed in my first slide, feeding back downwards onto the proteins and genes? One of those arrows is the electrical potential feeding back on onto these protein components. And if I interrupt that feedback, so I just cut it, which you can do very easily in a computation. You can also do it experimentally, though it's quite difficult to do. It's called a voltage clamp experiment. What you find is very striking. Not only have you abolished the electrical rhythm, of course you've done that because you've held the voltage constant, but you also abolish all rhythm in any of these transporters. It's an integrative process that can only be understood by putting it all together, first working out how the components work, and then integrating it together in the form of a model set of equations, and then solving for the process. For the technically minded and mathematicians amongst you, this really is a limit cycle. It doesn't have a stable point anywhere, because if you take put the feedback back, it simply starts to oscillate again automatically. My heart's beating rather faster than normal because I'm lecturing to you, and we know the reasons for that. There is a substance called adrenaline flowing through my circulatory system that upregulates all 3 of these transporters, and if we put that into the equations, we get a nice reconstruction of the acceleration of rhythm due to adrenaline. So if you fall in love or exercise or do anything else that produces adrenaline in your veins, this is what happens. And again, it's something you only understand as an integrative process. You don't understand it on the basis of the reductive view of the individual components. So what I want to do in the next 2 or 3 minutes is to show how we move up the levels I showed the levels. This is, of course, the cellular and protein level that I've reconstructed at the moment. About 15 or so years ago, I had the great pleasure to be a visiting professor for a while, right down under in Auckland, New Zealand, with Professor Peter Hunter. And what I found Peter Hunter and his colleagues were doing was extraordinarily tedious. They were taking a whole heart from an animal, preserving it, mounting it on stereotactic anatomical investigative instruments, and then measuring painstakingly the fiber orientations, that is the way the cells are organized around the heart, on the whole of that surface. Oh, so a few 100,000 points, and you store that into a database. You then shave a tiny fraction of a millimeter off, and you repeat the process, and you do that for the next 3 years. And if at the end of that, you've got about 50 million data points, which you can incorporate into an anatomical model of the heart. It's very accurate. I love to quote examples of research that would never be funded by the Medical Research Council, would never be funded by the BBSRC, and this surely is one of them. Peter Hunter had a vision. His vision was an e-science vision, in the sense is that it involved, first of all, arranging that database so that people like me could come along later and pour, as it were, our equations for the cellular activity into the anatomical model. And then make the two work together so that you could have electric excitation, contraction, blood flow, I'll show all of these in the next few minutes, occurring in a model of the whole organ. So you can go up, jump right up to the level of the whole organ, still retaining a large number of features of the simulations at the lower level. This is Peter Hunter and the Auckland ventricle model. And what I'm going to do, I hope, in the next slide, is to illustrate a more recent version of their, if we can get this to work, a more recent version of their work, which was done on a pig heart and done rather faster than in 3 years. And as I make this movie work, you'll see that we scan through the various levels, as it were, through the organ, rotating it through the model organ. And as you can see, all the various fiber orientations are nicely represented there. Remember, there are tens of millions of grid data points there, each one of which can be associated with the kinds of equations that I've shown you for the cellular activity. If you put all of that together, you can then reconstruct, for the first time, the spread of electrical excitation through a heart as it is being excited. So what we've got here is the anatomical organ, the tens of millions of grid points in the Auckland model, and then equations for the individual cellular components, and then the spread of electrical excitation as it exits from the wall between the two ventricles. I won't go through the details of what it's done before that, and then spreads out through the ventricular wall up and finishes right up at the back there. Incidentally, that process in your heart and mine would take about 20 milliseconds, so it's a very rapid process. To those of you who know the electrocardiogram, it corresponds to the very fast QRS phase of the electrocardiogram. So it's the very beginning of the excitation of the ventricle. 20 milliseconds, it took 36 hours on an 18-processor silicon graphics supercomputer. And even today, on some of the faster machines that the computing people have here in Oxford and in Auckland, it still takes a few hours. We're hoping to solve that problem. We have a contract with Fujitsu to develop the code necessary to run on the 10 petaflop machine that they're hoping to build in Japan in a few years' time. And we've calculated it would be possible to do these kinds of simulations in real time. Is it correct? Obviously, those who built models of aircraft, for example, for determining whether a new aircraft design would fly or not, had to test the models in the first place against experiment, and so we have to do in biology. Very fortunately, David Patterson's group in Physiology here in Oxford did experiments with 256 electrodes on a real, in this case, pig heart, and the electrical wave does indeed exit through the ventricular wall and then spread up and end up at the back. Very similar to the computation. What is constraining these calculations is, of course, the accuracy or not of the cellular equations, but also the accuracy of all the detailed imaging and anatomy that the Auckland team has done. We don't do that anatomy now so tediously. This is a lovely example, if I can get this movie to work, of scanning to get the anatomy of a whole organ. This is a rabbit heart, which was scanned. We're going from the top and now entering the two ventricles. There's the valves. We're now entering into the two ventricles. This is done at a voxel size of 25 micrometers cube, which is down at the cellular level. You can identify individual cells in the imaging here. Moreover, you can identify the courses of the blood vessels. Just follow any one of these blood vessels. You can see how it branches and forms the network. When you have checked this scanning information against histological checking of what biological types of cells there are there, you can build a three-dimensional model in much less than the 3 years that it took the Auckland team to do it in the first place. We're getting now down to the bottom of the heart. This is the very tip of the ventricle down towards the apex around here. That's one finished, and now we end up at the very bottom there too. What this raises is the distinct possibility that in the future what e-science of this kind can contribute to medicine is the possibility that you have scans of individual patient hearts and then transfer that into the simulations together with perhaps the disease state that the heart has or the drugs that are interacting with it, and then try to understand, if you can, the processes that must be going on in an individual's heart. So patient-specific information could be incorporated into these models, and indeed is already doing so. Here's a reconstruction of the coronary vascular system in that particular heart. How has that been used? Nick Smith, who is now here but was in Auckland, in fact, he seems to me to still commute between the two. You can't commute far further than that, I'm afraid. He's reconstructed the coronary vascular system in one of his models. This is a good example of such a model with a constriction here to represent what happens in ischemic heart disease, where an artery gets furred up to the point where there's a block of circulation. The blue area shows area that is deprived of blood flow. It's a lovely model, so I really must show you this particular model working. He has both the contraction properties of this particular model and the flow of blood through the vascular system, taking about 3 beats to flow all the way through. And he can even compute the interaction between the squeezing effect of the ventricle and the blood flow. That's important, obviously, for disease states, for reconstructing disease states that depend upon the flow of blood or the lack of flow of blood. We've reconstructed now very many forms of rhythm and arrhythmia of the heart. I'll show you just 2 examples that are relevant. One is a collaboration that Peter Cole here in Oxford, together with Natalia Tryanova in the United States, did to reconstruct a very particular form of arrhythmia. If you're unlucky enough to be on a golf course and somebody hits the ball and it hits you straight in the chest, you can fall dead. I didn't hit myself hard enough to do that. And I can tell you I cannot do it because I know that the energy that I've just used is nowhere near enough. We know the energy required, and it's about that of a golf ball or a hockey puck or something like that. And there are examples of people who are killed just by that process. They're not killed because of the damage. They're killed because of an interaction. The impact on the chest produces a little bit of compression of the heart. There are stretch receptors in the heart, proteins that respond to the stretch, which then generate electric current. And when you put those into the model, as you can see, stretch-activated channels are in this model, you get a beautiful reconstruction of that kind of arrhythmia. There's the impact just there, and here is a continually recycling wave of excitation. Instead of getting a beat like this, 1, 2, 3, thump, thump, thump. What you feel if this is happening to you is that. Very rapid tachycardia. You would fall down probably, you'd faint. You wouldn't be killed though. What kills is if that process breaks down, as it does in the next movie from Sasha Panfilov's work, a mathematician who's worked on the question, how do such reentrant waves break down? What are the characteristics of the kinetics of the protein channels that would lead to it breaking down, as it's now beginning to do, into little wavelets, so that the heart, instead of doing that, starts to just shimmer? This is what we call fibrillation. There's no further flow of blood, of course, when that happens. The heart, as it were, from a mechanical point of view, is almost frozen, and that's what kills. Incidentally, there's a recording experimentally on a real heart using a voltage-sensitive dye on the heart, showing a very similar pattern to the almost chaotic behavior that occurs when a heart goes into fibrillation. If you can understand that mathematically and computationally, then you can begin to do two things. You can begin to work out what is a better way of administering shocks to the heart. That's a defibrillator. It's what you see in BBC programs, Casualty or whatever it is, those great big electrodes that get brought up. The man jumps and hopefully his heart starts to beat again. You can do that better, and there are people like Blanca Rodriguez working on exactly that kind of question. Can we use the insights from these models to work out how to build better defibrillators? And you can also, of course, work on the question of what kinds of drugs would be better. Everything I've shown you so far has been on animal experimentation, followed by computation, and very recently we've put together a human cell model because enough information has now come, particularly from a team in Hungary that we're collaborating with, from cells that have been isolated during surgery on human hearts in surgical operations. I will show just one example of that where the equations have been incorporated into a model of the human ventricle, again showing the ability to reconstruct an arrhythmia of the kind that will produce that rapid tachycardia and possibly therefore break down into fibrillation. I want to finish the presentations. I want to leave a few minutes for questions and discussion. I'm going to finish the presentation with a movie which has a voiceover. It was produced, in fact, for the Millennium Dome. It was about Michael Heseltine and Tony Blair will forgive me for saying this. It was about the only part of the Millennium Dome that was a success. That was the body zone. And what was the great success in the body zone? Well, it was the heart model, and you'll see why. Let's see if we can get this to work.
The image you're looking at may appear to be a film of a living human heart, but it isn't. It's a highly sophisticated computer program using tens of millions of separate equations, combining graphics and mathematics to create one of the first of a new generation of simulations called virtual organs. These are set to become some of the most powerful tools we have in the fight against disease, with scores of uses from disease diagnosis and drug design to training and research. This program simulates the complex activities of the heart, and it shows us just how a real heart would react to a range of conditions, physical and chemical. Let's take a closer look. Inside a real heart lies the branching network of blood vessels which carry vital oxygen and nutrients to the rest of the organ. Each of these branches is precisely modelled by the software. The colours clearly show the flow of blood and how it changes with each heartbeat. The virtual heart can also show the electrical activity of the heart as it beats. Normally, electrical impulses spread out through cardiac muscle in a series of orderly rhythms. They are also represented here by different colours, showing the variations in the impulses second by second. Look at the difference when the virtual heart is simulating congestive heart failure, one of the major causes of illness among elderly people. Controlled electrical activity is being replaced by multiple overlapping waves that speed up the heart rate, just as can also happen during a full-scale heart attack. When this occurs for real, doctors and surgeons are too busy trying to save the patient's life to examine the progress of the heart attack in detail. But the virtual heart allows doctors to study the effects of a heart attack in controlled circumstances without endangering a real patient. And unlike real life, in in which a heart attack can take just a few seconds, the progress of the attack in the virtual heart can be slowed down, speeded up, and repeated at will, enabling scientists to measure exactly what is happening. Virtual organs such as this one allow researchers to develop and test new drugs, faster, more confidently, and at less cost. Scientists are already busy creating other computer simulations which work with each other to show the whole human body as an integrated dynamic system. This model of the torso, for example, shows how each heartbeat generates a wave of electrical activity that flows across the surface of the body as a whole. The virtual heart is just the beginning. Very soon, we'll be able to build a virtual model of the entire body and find better and faster ways to cure disease.
Well, very soon we will be able... You don't believe that, and nor do I. Maybe by the time scale of evolution, we will get there in maybe, 200 years rather than 2 billion years. But there we are. Thank you very much.
I'm going to take over for Paul Jeffries, build up, and Dennis, that was excellent. I mean, you really focused on the computational changes in computation from 1960 to now and how it's changed the sophistication of simulations. But could you say something about the collaborative? No, you said 200 people. In 1960, you weren't using Web 2.0. Probably not now.
No.
What were you doing in 1960 to collaborate with whom, and how has that changed?
Well, there was only one of me in 1960, and of course Alan Hodgkin and Andrew Huxley to talk to, and they were in Cambridge, and that kind of conversation was actually quite rare, though Alan Hodgkin did examine my thesis. I think a big element in what we've done is first, management, and second, using to the full all the modern means of communication. You won't be surprised, therefore, that internet, video conferencing, and all the rest of it is part of what we're doing all the time. There would be no way of collaborating with the people right down there in New Zealand without using all of the facilities. And that's one element. The other, though, I mentioned was management. It's actually quite difficult to bring teams like this, interdisciplinary teams, together. The mathematicians, you see, they love to publish papers on their own. Have you come across a mathematician who doesn't, have just one author at the head? Well, we have to train them, you see, to recognize that these are collaborations, and you have to take part in a great big collaboration. The engineers, they have a different concept of what it means to do the kind of work we're doing. So we have to introduce them to the biology. I think the key to what we've been able to do has been that 2 or 3 of us, myself, Peter Hunter, Ray Winslow, and Nick Smith, and 1 or 2 others, have, as it were, recognized each other's strengths and talents and managed, as it were, to manage them together. And management is a key feature of being able to build networks of this size.
Okay, unfortunately, we should probably move on. You can see from the agenda that we're trying to use a limited number of speakers, limited number of time, not only to tour disciplines, but also to tour different foci of e-science. Actually, Dennis's talk really illustrated the visualization potential of computation. But Martin Kemp will be speaking next, and we'll move to the humanities, and I actually think visualization will come up even more powerfully.
Well, whether this is going to be history of art or not, you can make up your own minds. Who cares is my answer to that question. I've been working with the modeling, visualization and modeling of visual phenomena on computers since the late 1980s, so quite junior compared with Denis in that respect. And the first time I did it was for an exhibition on Leonardo da Vinci at the Hayward Gallery in 1989. And we worked with the IBM Research Center at Winchester, and we modeled some of Leonardo's ideas. That is to say, put the basic notions which Leonardo is working within relating to light, branching systems, geometry, church design, and so on, and let them run initially, the IBM Research Centre people thought this is going to be simple, but after 6 people had been sweating on it for 6 months, they realized that it wasn't quite as simple as it looked. I'm not going to be showing you those, but I'm going to be showing you 2 types of work I've been involved with more recently. One is on space on paintings, and anybody who knows my research and the work on optics and perspective in the visual arts will not be surprised to find that part of it is on space. And the second part is to pick up on more recent modeling of Leonardo. Essentially what I'm doing is visual modeling. The prime purpose for it and where the funding comes from is for public exposition. The funding doesn't come from research councils. Indeed, I couldn't even go to the Arts Humanities Research Council with this, I know. It comes from television programs and it comes from exhibition budgets, oddly enough. At the end, we'll get onto an area which is very close to Denis, and I've chosen an example which relates to the human heart in Leonardo. Anyway, let's see where we go. The examples I've chosen are familiar old war horses of perspectival analysis, Renaissance paintings. This has been undertaken in collaboration with Antonio Cremonesi, who was initially in the robotics group in engineering here, doing his doctorate in computer vision, now working for Microsoft at Cambridge. And if anyone wants to follow up the joint papers, they are available on his site, the Microsoft site at Cambridge. His name is Antonio Cremonesi, Italian as the name suggests. The great advantage of working with an Italian is you don't have to convince them that analysis of works of art is essentially interesting, which doesn't come naturally to all people in the computing field, I have to say. Anyway, the Masaccio Trinity, a heavily analysed work. I analysed the perspective of this by hand, the normal way, drawing what I call fat lines on small reproductions, with all the resulting imprecisions that come in with this. Now, Antonio, for his doctoral thesis, Antonio Cremonesi, devised a technique called single-view metrology, which basically can take any photograph or systematically designed perspectival image and go from the image into 3 dimensions without any CAD, without any computer-aided design system involved. It was developed initially for forensic purposes, for photographs of crime sites and so on, and he can do it without knowing the position or the parameters of the camera. It's a smart, very smart system. It uses something else that was developed in the computer vision place here called a canny edge detector, and there are now much more advanced ones, but I always liked the name canny edge detector. The man who invented it was called Canny. It's not because it's clever. Some people follow their names rather nicely. And with Antonio, we looked at some of these classic cases and saw what happens. And basically, you have to obviously take some starting point as to one item in that painting, one object you assume is a regular given shape. You need a starting point, but once you do that, and with his program, you can go straight into the analysis. You can produce, and these are stills from a fly-through that we did. There is no visual data here other than is extracted from the surface of the painting, hence the figures remain flat, and we didn't render the occluded areas, the black areas and the vaults there, and you can produce a very nice fly-through. This is chiefly about exposition, but of course it does tell you a terrific amount about what is in the painting, its internal consistency, what went into it. In a sense, by what we can get out of it is only related to what the artist put into it. And this is probably in 1427, the first systematic perspective painting of the Renaissance. And it is astonishing how spectacularly it holds up to this, that its internal consistency is remarkable, though, as we shall see, there is one point at which it is open to doubt. What we found is that there are 2 obvious assumptions you can make about the Trinity. The first is that the coffers are square, and the second is that the ground plan of the building, the notional building behind the picture plane in which the figures exist, is square. It turns out these 2 starting assumptions are not consistent with each other. you have to choose. On the left, there is the symmetrical, totally symmetrical, very nice sort of Renaissance style centralized structure, looking down on it from the top and from below. And once you look at it from below, you can see, even I think just visually, that the coffers come out non-square. As soon as you render the coffers square, the building comes out non-square, which meant that somewhere along the line there is an inconsistency in the design process there, and the computer program, the computer analysis won't tell you which choice to make, obviously. It's really a question of going back to the picture and using historical judgment as to say, is it more likely that Masaccio, the painter of this, began with a square ground plan or with square coffers? It might look obvious in a modern sense that the square coffers are the obvious place to begin, but if you think about putting a building in perspective, that is to say projecting a ground plan into perspective, that's the point you begin. You don't begin with the detail, you begin with the overall configuration. And I think it's fairly clear that he intended his building to be this remarkably harmonic, centralized structure. And he was prepared to tolerate the fact, even if he analyzed it, that the coffers would go off square. They look fine. But it's a matter of historical judgment. So in a way, this is exposition, but it always along the line you come up with questions you haven't quite asked in that sense before. The second of them is an even more famous warhorse of Renaissance perspective, the Piero della Francesca flagellation. For those of you who haven't seen the picture in the original in Urbino, it's 58.3 centimeters high. It's a small picture, which will give you a sense of the kind of precision with which Piero della Francesca is operating. A picture from the 1460s, just to give a rough location in it. This is obviously a wonderful field day for single view metrology. Once you've made one assumption about a square tile, which you can reasonably well do, you end up with all sorts of exciting things coming in. I'd previously teased out by hand, as it were, the basic tiling pattern. This tiling pattern which exists here in front of Christ, he's standing, being flagellated on a round tile, a round dark tile inscribed in a square. And this pattern here is very interesting because the diagonal of the shorter squares is the side length of the larger squares. That's to say it's a root 2 pattern, which is quite a smart pattern. What we came up with Antonio's system, and I'm sure, sorry, this is actually a relatively low resolution image. There are better ones, but confirming this basic pattern, though there's some doubt as to whether Piero's got that area quite right. These are all the occlusions, which we don't put in. We could easily move bits of rendering across if we wanted to do so. But at the back, what you could more or less see there, and I'm sorry, this is not a good resolution image, is that the tiling pattern back here is also rendered accurately. And that on that scale is just awesome because you're getting to the point where the lines you draw are almost as fat as the spaces between them. So the levels of precision which Piero is operating with in this small painting is just awesome. And I hadn't by hand, though I worked with a full-scale reproduction. I actually got a proper one-to-one reproduction of this painting. I'd not got out that at all doing it by hand. I suppose if I'd persisted enough, I might have done so. We can do other things very readily with single view metrology. We can simply measure heights, which may seem a bit tedious, but it does indicate the extent to which Piero is trying to be consistent in the scaling of his figures in relation to their place in the painting. This man is the only one who is taller to any notable degree, but even that's hardly significant. But you can see, I think, why Christ is made to look quite vulnerable against the slightly higher man. This man wears a hat. So all the time there are artistic decisions going on about what looks best in terms of the narrative. These are not simply works of perspective geometry. The other scalings are extraordinarily accurate. So Piero has obviously calculated these figures in relation to their relative scaling and the tile patterns. Again, this is, it shows you the kind of perfectionist Pirot is. He painted very few pictures. Surprisingly, he is in the job of slow art rather as like slow food. And the more you look at that, a figure who's as obsessive with getting things right, that the interplay with this powerful tool of analysis reveals that very clearly. The third one, there's a movie in there which I'm not running as a movie, is the painting of the National Gallery in London, the Paolo Uccello Rout of San Romano, one of 3 panels painted of the famous Florentine battle, probably from the 1430s. And this was for a television program, The Private Life of a Masterpiece. And as I say, this is where the funding comes from. In the arts, the television program, particularly the American transmission, gets generous funding, the kind of funding which I simply can't get hold of for other purposes. And if I build physical models of, say, Leonardo flying machines, we do that with TV money. We don't do it with research money. Anyway, I knew from the analysis of the Uccello hunt, which is in the Ashmolean here, that even under pictures without tiled floors, Uccello created a kind of ghost tiling pattern. We got that out with infrared reflectography from the Ashmolean hunt. And I was pretty sure, though the National Gallery people didn't believe this for a moment when we started, that this picture here, with its rather carefully designed, these broken lances fall unerringly in a kind of semi-perspectival order, that there would have been a ghost pavement under here which would have provided for the scaling of the figures. Antonio and I reconstructed that ghost pavement. There are some significant diagonal elements which actually give you the size of the tiled square, and it comes out, if you do that, in what is a very standard Renaissance perspectival construction with the central vanishing point and the so-called distance point here. I won't go into the technicalities of that. Anyway, for the purposes of the television program and as a kind of verification, Antonio deprojected the floor on which this detritus lies, and you can see how many, the significant number of them are actually really rather nicely parallel. He's pumped up the size of the more distant lances to make them apparent. Again, he's not followed consistency, he's made it look right visually. And in fact, there are the 2 diagonals going up here, another one here, which are the ones which show the tiled floor. The next step of this research would be to persuade the National Gallery to do a full X-ray infrared reflectography, full imaging analysis of the underlayers of this picture, and I would bet them a lot of whatever I bet them, that out would come a ghost tiled floor. And for the television program, we did a little animation of moving the central figure. This is the Florentine commander here, this man with his rather grand hat on. Incidentally, the hat would have a mazzocchio, a faceted structure inside it, which Uccello also worked out in terms of perspective drawing. And we slid this figure backwards and forwards to check the scaling at various points. When I mounted this little animation, I found that it's a very low resolution and very fast, so catch it while it goes past. This was for the television program, really as a bit of public exposition. But what was nice in this context was that the hunch I had that there was an order, a ghost order, as it were, in this chaotic battle scene, deprojecting it did stress that a significant number of the items are actually working rather systematically in relation to the notional tiled floor, which is used to structure the scaling in this case. The fourth spatial example is more surprising, and when we started, it was done as a kind of game, and the results are potentially revolutionary. And this was looking at a number of convex mirrors in Netherlandish paintings in in present-day Holland. And a number of them, we obviously did the Jan van Eyck Arnolfini wedding in the National Gallery, which is perhaps the most famous of all these convex mirrors. But the one which yielded the most spectacular results was in this painting in Madrid, which is the left wing of an altarpiece. The central wing has disappeared. Obviously, you would have the Madonna and Saints in the middle, commissioned by a Franciscan, Canon van Verl, who is kneeling here with John the Baptist and this very splendid window in the background. Netherlandish artists, rather like Perrault, worked very small, and you can see the surprising amount of detail that there is in this convex mirror hanging in the wall, the room divider, in the background of this painting. Antonio produced algorithms for rectifying the mirror. We produced ones for parabolic mirrors, just to be sure, for mirrors of various bulge, and you can work out with trial and error the relative bulge of this mirror. And we then rectified it. And what came out was that, just rectifying it with the algorithm, the rendering in that mirror is just staggeringly good. The extent to which the straight lines come out straight is uncanny. The Jan van Eyck Arnolfini wedding also works very well in that respect. Now, you don't do that by guesswork. And also what we began to notice was how brilliantly the occlusions are observed. That's to say here, and again, it's not terrifically good resolution, the drapery of Van der Verl behind this door is rendered, and you can't see him behind that door. He's somewhere in front of it, so you can sort of work out what would be occluded. Now, if I gave you the task of putting a mirror on a room, just thinking about it, and working out the occlusions with a flat mirror, that is tough. If I asked you to do it with a convex, a curved mirror, I don't think anyone, however good they are at visualizing, could do that. And the implication for me is that the only way you do this mirror as brilliantly as that is that you actually have to have this scene in front of you. You actually have to do it from the mirror. He's got actors dressed up as John the Baptist. Van der Verl, he's taken his portrait, but probably has somebody kneeling in his place. And interestingly, there are some other Franciscans lurking in the back of the room. This man is perfectly scaled, this man is a bit small. Again, we've done scaling on that. And what this tells the people in Netherlandish art, and they don't really want to know this, is that the way these things are done is basically by tableau vivant. This would be regarded as old-fashioned naive art appreciation to say it looks very real, but the reason this looks very real is because it was very real. It was done, it must be done, I think, and I don't see how you can get around this with an actual mirror, an actual setup, with figures posed in this way in an actual room. And that, I think, you can carry through to other Netherlandish paintings. And the people working in Netherlandish art, rather like the people who I've done work with David Hockney on Vermeer and so on, art historians become very twitchy when you tell them they've basically got their artist rather wrong and have missed the main working method along the way. So this is not likely to have a particularly good reception, I think, in the first instance. And that has further potential, I think, in terms of looking at things in paintings which wouldn't be got right unless you actually had a real physical setup in them. And it obviously refracts into the David Hockney hypothesis, which I've been working on and modelling on with him in various other ways. His hypothesis that optical instruments have been used very heavily in the construction of these paintings. I don't think, incidentally, an optical instrument has been used here. I think it's just brilliant what David would call eyeballing. Two Leonardo incidents. One is purely expository, and these are both taken from the recent Leonardo exhibition at the Victoria and Albert Museum, which closed in January this year. And we worked with Cosgrove Hall, or Steve Meyer, who was then working for Cosgrove Hall, is now set up on his own a firm in Manchester who do many of the leading British animations. And we were animating some of Leonardo's ideas to make difficult things apparent to the audience, and secondly, to give a sense of Leonardo's own modes of visualization. Leonardo clearly could do moving 3D sculpture in his mind. He could do it by thinking about it. He was one of the people like Kepler who basically could model things in their minds and can see things in 3D, can manipulate them. I won't go into all the evidence for that, but perhaps you can just take my word at this stage that Leonardo's drawings show absolutely that he can do that kind of modeling. As I say, the first one is mainly public exposition, to give you an idea of the motivation for doing these. And in the exhibition, we had this illustration in the top right-hand corner of a truncated and stellated dodecahedron shown in skeletal form. And Leonardo invented this mode of portrayal in skeletal form as well as full plastic form for a manuscript then published as a book in 1509 by Luca Pacioli, his mathematician colleague, called De Divina Proporzione. Now, in an exhibition, it's terrifically difficult in a short label to explain to a member of the public what a truncated and stellated dodecahedron is. And you're likely to lose your audience, however enthusiastic they are, very early on. So what we did was to produce an animation. And this is based upon one of the codices in the Victoria and Albert Museum itself. I'll just let it run. Whether you'd start with an icosahedron or a dodecahedron makes no difference in terms of the finished product. And that's aimed really at what I call the aha factor. That looking at the book and then looking at that, you can see exactly what's going on, and people who thought they'd never understand how that object came about did so without great difficulty. And we worked, of course, very hard to get the color of the ink right, the contrast right. These were shown with Leonardo drawings, which is dangerous. You can easily be accused of crude Disneyfication of Leonardo. That didn't happen, as we really sweated with Steve Meyer in getting the visual texture, as it were, of these to be good-mannered in relation to the Leonardo drawings. Now, the second one brings me into Denis's territory. And I'm going into it historically, and I'm absolutely, well, actually, there is a bit of contemporary art science in this as well. One of the drawings I've been fascinated with over a period of time are these late drawings in 1513 or so by Leonardo of the heart. This almost certainly is an ox heart, though he also did have access at this phase to human material. And he became terrifically interested in, there incidentally is the coronary artery. He became terrifically interested in these three cusp valves, the pulmonary aortic valve, and saw it as a piece of living geometry, the kind of transformational geometry we saw earlier. This is transformational geometry in the human body. And he isolates the valve visually, as it were. It's just terrific. Nobody did anatomical drawing like this. The valve isolated from above, the valve with its cusps open here from the bottom, seen from below, open, and open from below. Amazing sort of plastic demonstration. We take this sort of thing for granted now, but really Leonardo invented this mode of demonstration. And he was interested not just in the geometry of the valve as it opened as a kind of piece of transformational geometry, but also in visualizing the flow within the valve. And to do that, he brought his studies of water movement to bear upon these phenomena. This is a study he made of a weir here with a hole and these spiraling vortices. And up here, he says, make a model in glass, a prova, as he calls it, a test in glass, to see what happens. Now, I was contacted while we were in the run-in to the exhibition when I was in America by Maury Garib, who is Professor of Aeronautical and Biological Engineering at Caltech. Wonderful thing to be, Professor of Aeronautical and Biological Engineering. Leonardo would love to have been that. And he was talking about building a model. So we had a dialogue, and we have actually published on this, about Leonardo's designs for this heart valve and his model. And Maury built initially a model of the neck of the aorta and eventually put the valves in it. And the valves are entirely passive. Denis may tell me otherwise, but anyway, the valves basically are working in a passive way. And this is looking down on Maury Garib's model, and here are these flaps, the flaps of the valves here in a flaccid state. Here's Leonardo's drawing, and using modern imaging techniques, this is before we've actually put the flaps in. This is just looking at the behavior of the neck of the aorta. Here we've got these vortices which fill the cusps, and they fill the cusp before the next pulse of heart blood comes up from underneath, then filling them again. And this basically is the operation of the valve. So here we've modeled Leonardo's valve, not involving computers here, but involving physical models. But obviously the imaging technique is fed through the computer. Leonardo couldn't have ever seen what happened inside the heart valve. We obviously now can model it, and indeed we can see inside the heart, we can image the actual vortices in the heart. While this dialogue was going on, I was also in dialogue with Francis Wells, a leading heart surgeon at Papworth, who was again re-looking at the Leonardo drawings. And I put Maury Garib and Francis Wells in touch with each other, and they have been working anew upon the dynamics of flow in the mitral valve and in the tricuspid valve, and are publishing in this area. And the end result of this in the exhibition was that they both fed into what was then the demonstration of Leonardo's heart valve or heart in operation. Keep your eye on these little fellows over here. And that's then into the weir design, which you've already seen. This is a little story in a way where something which I thought of as simply a way of public exposition has all this extraordinary spin-off when you talk to people in other areas. And Maury Garib and Francis Wells, as I say, are working actually together. Francis now has, as a result of the imaging of flow in the mitral valve, a new operation, which his patients call the Leonardo operation. So in these areas, once you collaborate in this way, you never know where you're going to end up. Thank you very much.
Thank you, Martin. I mean, what a beautiful example of how e-research actually changes the way we view art, but not just change the way we view art, but what we can see. So it actually changes the outcome of what we do, not just the process, and it's excellent.